Hi, my name's Blair and I'm from the Faculty of Science at the University of Technology in Sydney. And with this vodcast, we're looking at the subject cell biology and genetics. And this is part 3 of a 3-part series on proteins. This presentation may contain material that is protected under copyright. You may not copy nor distribute any of this material. This presentation can only be used for your own personal education and learning. So this is a 3-part series on proteins. And by the end of the 3rd part, you should be able to describe the characteristics, structures and functions of amino acids and proteins. And by end of this vodcast, part 3, you should be able to understand and be able to describe the relationship between protein structure and function in relationship to disease using appropriate examples. Our first example is sickle cell anemia. And anemia is an error in red blood cells. And sickle refers to the shape of the red blood cell. A normal red blood cell is a donut shape. It's round and it has a slight hollow in the centre. But sickle cell red blood cells become elongated and flat. They lose that hollow in the centre of the red blood cell. Because of this strange shape, sickle cell red blood cells have a decrease of oxygen transport and also can lead to clots in blood vessels because the altered shape changes the way in which they travel through the blood vessel. Sickle cell anemia comes about due to an error in hemoglobin. Hemoglobin is a tetramer, and it is a heteropolymer. It's a heteropolymer because we have 2 alpha chains and 2 beta chains coming together to make one super macromolecule. And hemoglobin carries oxygen in your blood. Sickle cell anemia is caused by a single amino acid change. And remember that with proteins, sequence gives structure, gives function. We have a single amino acid change in which a valine, which is a hydrophobic amino acid, gets situated on the outside of the fold. Now that this valine is on the outside of the protein, it has to interact with water. But valine is a hydrophobic amino acid and doesn't want to interact with water, which is polar and hydrophilic. So therefore, these valine residues interact with each other. And this causes the molecules of hemoglobin to come together and form strands. These strands sit in the cytoplasm and push out the sides of the cell, which gives us that sickle shape. P53 is known as a tumour suppressor protein, and the tumour refers to cancer. So P53 is an anti-cancer protein. And P53 interacts with DNA. So here we have our P53, which is our protein, and it interacts with DNA. DNA is our DNA is our genome. And within P53 are amino acids, such as R, which stands for arginine. And these different amino acids interact with the DNA. If these amino acids are changed, we lose that interaction with the DNA. The job of P53 is to stop the cell cycle. So we have here a cartoon of the cell cycle where this passage is moving towards DNA replication. The job of P53 is to stop this process, stop the movement to DNA replication until any mutations are repaired. If P53 doesn't work, the cell cycle can continue and therefore mutations may be carried to daughter cells. The build-up of mutations, errors in DNA, is associated with the development of cancer. Cystic fibrosis is an error in iron transport. And a classic symptom of cystic fibrosis is an increase in mucus in the lungs. It's a single amino acid change, a single mutation in the CFTR protein, which is the cystic fibrosis transmembrane conductance regulator. And this single amino acid change is phenylalanine. This single amino acid change is due to a single mutation in a codon. And a codon is 3 nucleotides. This phenylalanine mutation alters the sequence, which therefore alters the function. The function of CFTR is as a chloride ion channel. It moves chloride ions from inside the cell to outside the cell. When this protein is mutated, that is one amino acid change, we lose this movement. Because the chloride ions don't move, we have a decrease in the movement of water, which increases the viscosity of the mucus or the thickness of the mucus, and this leads to an increased risk of bacterial infections. Prions are infectious proteins. They lead to formation of a spongiform encephalopathy. An example of an infectious spongiform encephalopathy is bovine spongiform encephalopathy, also known as mad cow's disease. The way in which prions work is taking a normal protein, which is an alpha coil, and converting that normal protein to mostly beta sheet. This abnormal or infectious protein aggregates in the neuronal cell. This aggregation within the neuronal cell leads to formation of plaques. These plaques look like holes. And where we've got these plaques, it leads to neurodegeneration. This neurodegeneration leads to a dementia-like illness. So in summary, for part 3 of our series on proteins, sequence gives structure, gives function. Alterations in sequence can alter or knock out the biological function of a protein. For example, we looked at sickle cell anemia, which is a mutation in hemoglobin, P53 and its relationship to the cell cycle and cancer, cystic fibrosis and the CFTR transmembrane protein, and prions, which are infectious proteins. Now the take-home message of our 3-part series on proteins. Sequence gives structure, gives function. Proteins are polymers where the monomer is the amino acid. Amino acids have different physicochemical properties based on the character of their side chain, which is also known as an R group or a functional group. Proteins fold, that is, they have a 3D shape, by hiding hydrophobic residues on the inside, away from water, and leaving the hydrophilic residues on the outside so they can interact with water. This is called hydrophobic exclusion. Proteins also are stabilised in their 3D shape by, for example, ionic interactions between charged amino acids and hydrogen bonds between polar residues. Misfolding, that is, an alter in the structure, can lead to disease states.